Hello dear students, Assalamu alaikum. I am here with the lecture number 2 of the embryology series, and this lecture will cover the fertilization, cleavage and the formation of 3 different germinal layers. I am Dr. Muhammad Shuaib Khan, PhD in Anatomy from Universiti Putra, Malaysia. I am lecturer in the Department of Basic Veterinary Sciences, Faculty of Veterinary and Animal Sciences, Gomal University, Deir Ismail Khan. I am very thankful to my fellow colleagues Dr. Sidra Mariam, MPhil Histology, and Dr. Ziaullah, MPhil Anatomy, for their help and contributions in preparing this lecture series. In the previous lecture, we have learned about the histology and embryology definitions. We have also learned the process of gametogenesis. We have also learned about the process of spermatogenesis and then the oogenesis. We have also discussed in detail about the folliculogenesis. The folliculogenesis lead to the formation of ova, or furthermore we can say that the folliculogenesis is the process leading the process of oogenesis. So don't confuse between the folliculogenesis and the oogenesis. This process ultimately lead the formation of ova and then the fertilization, and if the animals not fertilized at that time, if animal not become pregnant, the cycle, or we can say the estrous cycle, they begin again. So today lecture we will focus about the process of fertilization. We will start from the capacitation and then we will end on the formation of germinal layers. This slide is just to memorize you about this process of spermatogenesis and oogenesis. You can see the diagrams we have already discussed, that is the process of the spermatogenesis, mitosis 1, mitosis 2, and then the differentiation and the formation of the sperms. So primary spermatocyte formation, secondary spermatocyte formation, and then the spermatids nourishment from the sterilized cells, and then the sperm formation. So this is again we can see the process of folliculogenesis, and the process of folliculogenesis ultimately lead to the formation of ova, and you can see the primordial follicle and then the primary follicles, then the early secondary follicles, and then the maturing follicles, and then the mature follicles, and after this the formation of the corpus luteum. We also discussed the formation of corpus luteum and the role of corpus luteum in the previous lecture. You can if you don't remember, you can first go to the previous lecture and hear about the what the corpus luteum do actually, and what if the corpus luteum not form and the corpus luteum degenerate as the corpus albicans. So I previously told you that the corpus albicans, if the pregnancy not occur, the corpus albicans is the structure which is leading to the formation of starting of estrous cycle again, and then the formation of the primordial follicles. In the estrous cycle, animals show the heat signs. Usually dairy cows become restless, or the buffaloes or the other animal, they become restless, they produce the bellowing sounds, they mount on the other fellow animals. So the farmer detects that this animal is in heat. So what he do? He go for the two options. Either he have bull in his dairy farm, so he use the natural service, or he call the inseminator, the artificial inseminator, to inseminate the cow. The artificial insemination is a common practice nowadays, doing like adopted by the farmers. So what happen? Usually the capacitation occurs after the ejaculation. If you are doing the normal service of a dairy cow, that the spermatozoa they usually deposited into the vagina. So when the spermatozoa leave the vagina and enter the superior female reproductive tract, here the capacitation occur. The capacitation, we can say shortly that this is the maturation of sperms inside the reproductive tract of the female animal. This is the step of a biochemical events The sperms move normally and look mature prior to the fertilization. So capacitation is the step in the maturation of mammalian spermatozoa and is required to render them competent to fertilize the ova or oocytes. According to the Encyclopedia of Reproduction, 2nd edition 2018, the capacitation causes a series of signaling biochemical and structural remodeling events that release the spermatozoa from oviductal reservoir site, remodel the sperm surface endospermatozoa with hyperactivated motility, and prepare them for interactions with the oocytes at fertilization. So in vitro capacitation and fertilization. In vitro means inside the test tube. Don't confuse when you read these two terms, in vivo and in vitro. In vivo means inside the animal. In vitro means inside the test tube. Sometimes some people use another term, that is the ex vivo. Ex vivo means that when you experiment or do some experimentation outside the body of the animal. Like sometime we use the biopsy. So the biopsy is a kind of ex vivo work. Sometime you take the blood of the animals and you do the other experiment outside the animal body or in the laboratory. So this is the something about the in vivo, ex vivo and in vitro. Students become confused at the end usually. Even they pass their degrees and they don't know about these three terms. So these are very simple: in vivo, ex vivo and in vitro. So in the process of in vitro fertilization, capacitation occurs by incubating spermatozoa that have either undergone ejaculation, extract from the epididymis, or incubated in a defined medium for several hours. This is about the in vitro capacitation and fertilization. Before coming to fertilization, I tell you something very important, that the ability of dairy cows and buffalo to successfully mate, conceive, giving birth and raising a healthy calves each year is very essential for the countries having a belief that they will bring a white revolution, that they will produce some more meat, that they will produce more milk. And for you people who want to pursue their career in veterinary medicine or in animal sciences, that a good understanding of anatomy and physiology of both male and female animals will help you to successful management of these animals, which will ultimately lead to the production of good quality of beef and good quality and quantity of milk. So what is fertilization? Fertilization of union of a haploid oocyte and a haploid spermatozoon producing a diploid zygote, i.e. cells capable of developing into new individuals. Fertilization begins with the gamete fusion, zygote formation. Fertilization ends with the initiation of zygote cell division, i.e. the start of the cleavage. So these are very important points. These 2 points are very important. Fertilization begins within the gamete fusion. So the first step of fertilization is the formation of gametes, and the end of the fertilization is the start of the cleavage. So fusion of spermatozoon with an oocyte take place the uterine tube, or we can say the fallopian tube near the ovary. The spermatozoon must bind to a specific glycoprotein on the zona pellucida surrounding the oocyte. This species recognition process prevents unions with the foreign sperms. One sperm fertilize the single ova. If many sperms interacting an ova, this is a disease condition called the polyspermy. So usually what happens that only one sperm in the million of sperms only capable to fertilize the single ova. So what happened next? The spermatozoon releases degenerative enzymes acrosomal reaction. The enzyme denature the zona pellucida, allowing the sperm cell to penetrate the barrier. The spermatozoon and the oocytes plasma membrane they fuse. So the secondary oocytes complete the meiosis here. The oocyte immediately cancels its membrane potential via calcium influx, and then the denature is zona pellucida where enzymes are released. Male and female haploid pronuclei make contact, lose their nuclear membranes. Mitosis begins here just after 12 hours of the sperm's fusion to an ova, and the DNA takes place before the mitosis. So I previously told you that to know these all things, you should know about the anatomy and physiology of female reproductive tract. The oviduct begins as a funnel-shaped tube that engulfs the ovary. The funnel portion of the oviduct is called infundibulum. In the next slide, in next lecture, I will in detail tell all about the anatomy of the female reproductive system. So when the ovulation occurs, the ovum is picked by the infundibulum channel into the oviduct, also known as fallopian tube, where the fertilization takes place. And if viable sperms are present, here the ovum remains capable of fertilization of only a short time. Thus, it is essential that the sperm be present in the oviduct near the time of ovulation. For this phenomena, capacitation is very necessary. This capacitation, this make a sperm capable of going to that specific place. The ovum moves through the oviduct into the uterine horn with the next 3 or 4 days. If the ovum becomes fertilized, then so in the previous slide we were discussing in detail about the fertilization. So we were there that if the fertilization occur, corpus luteum become mature, and this corpus luteum is responsible of releasing the hormone of pregnancy. And if the corpus luteum degenerates, if the pregnancy not occurs, if the fertilization not occurs, this corpus luteum degenerates and end with the product names as the corpus albicans, which is a yellow pale structure, which end usually the estrous cycle and start the new estrous cycle. So we usually divide the pregnancy into the 3 trimesters: the first trimester, the second trimester, and the 3 trimester. In the dairy cows, the gestation time usually 39 to 40 weeks. These are the very important information. Just I realized that I have one slide about this essential information during that lecture of the fertilization. So the fertilization age at the moment 12 to 18 hours after showing the signs. So what usually we ask the farmers, what usually we teach the farmers, that service the cow or call the artificial inseminator just over the 12 to 18 hours. That also the sperms also find as necessary period for the capacitation. So divisions of pregnancy corresponding to the development: 0 to 3 weeks, this is the time for the early development; 3 to 8 weeks it is called the embryonic period. Organogenesis usually occurs 8 to 9 weeks. This is called the fetal period, and the total gestation time in some books there is variable. So some say 39. So we can also say that 39 to 40 weeks are usually the pregnancy time in the dairy cows. We are talking about the dairy cows. Each animal having its own time. This is another slide. Usually I made it for the students that the things make easy. These are the some important steps you have to remember. This is the week 1. What happens after the fertilization? Day 1 to 7 in dairy cows. So in the fertilization it usually happen at the day first. The cleavage, it start from the ultimately just after the fertilization and end by the second to third days, and then the capacity, the compaction for the day third, and then the formation of blastocytes day 4 to 5, and this process ends ultimately with implantation of fetus that happens usually on day 6 or 7. So this is the period for the dairy cows. Maybe the buffaloes having the some little bit differentiation. Also some other animal like camels, or maybe the giraffe, or the some other animal, they may be having their own time periods. So little bit variation depend upon the species to species. So it is a detailed diagram. This diagram is taken from the Human Embryology and Developmental Biology 4th edition by Carlson. So the you can see the evolution process, and then the fertilization. You can see here the process of fertilization, and then after the fertilization, the movement of that fertilized zygote. Here we can say the zygote formation and movement of zygote inside the fallopian tube or uterine tube. So at the day first, the cleavage occurs, then the formation of morula, then the formation of the blastocytes. They usually ends at the day 4, and then the implantation. So implantation usually occur at the day 6, and implantation occur in the horns of the uterus. In animals, maybe the left or the right horns of the uterus may be the gravid after the implantation. So if there are twins, both horns will be gravid. So if some animals they produce more than 5 or 6 babies, like especially in bitches, so some implantation occur in the right horn, some occur in the left horn. So both horn, in case of the more than one babies, they become gravid. This is another diagram showing in detail all about the fertilization, that how a single sperm fertilized an ova. You know, in one ejaculation there are many millions of sperms, but only one sperm that become capable of interacting the ova and fertilize the ova. So what usually happen? So previously we told you that how the sperm enters into the space that is called the corona radiata, and then it interact the inner acrosomal membrane, and then it pass the inner acrosomal membrane. I previously told you that the calcium ion influx that is responsible for interacting here in the membrane membranal interaction. So then the zona pellucida. So the sperm enters into this zona pellucida and cross the zona pellucida and enter the previtelline space. So you can see that finally the sperm also crossing the previtelline space and enter into the plasma membrane, and then finally they cross the plasma membrane. So this is a detailed and very nice diagram illustration of how the fertilization occurs. This is another illustration. You can see showing that how the sperm interact with the ova and how the fertilization occur. So you can see the detail that how the sperm head called the acrosome, they interact with the ova, how it enters, and then the actin formation, the actin filaments, they enter into the vitelline layer, then the acrosomal process, how they fuse into the plasma membrane, the pre-vitelline space entering. Usually this hyaluronic acid, they have also a role. This hyaluronic acid, they protect the sperm into the ova. So here what they do, that they produce an enzyme, the sperm hyaluronidase enzyme. So this fusion become possible here, that the sperm enters into the zona pellucida, corona radiata, and then the vitelline space, and then the previtelline space, and perivitelline space, sorry, we can say. So just to remind the events to you, that during the estrous cycle, a single egg is just capable of ovulate the ova. So this single egg is ovulated and swept into the oviduct. Fertilization site, this usually occurs into the ampulla, into the ampullary isthmus junction. Don't forget this name. Where the fertilization occur, in the ampulla, the ampullary isthmus junction. This is the fertilization site usually inside the in the domestic animals. So usually it occurs in the ampulla of the oviduct within the 24 hours of ovulation. So what happens after the fertilization, that this series of a cleavage and differentiation events results in the formation of blastocytes by the 4th to 5 days. The embryonic day depending on the species of animals. I previously told you that the variation is there in the data I given you. In the buffalo maybe the data sometime different than the cow, or in the camel the data sometime more different from the dairy cow. Same case in the horse also. Animal, the inner cell mass generate the embryonic tissues, outer trophectoderm generate the placental tissues. The implantation into the uterine wall occur into 0.6 to 0.7 days after the fertilization. It also depend on the species to species variation I previously told you. So now coming to the cleavage. The cleavage refers to the initial series of mitotic divisions by which the large zygote is fractionated into the numerous normal size cells. So each daughter cell of the cleavage process is termed as a blastomere. Just go to the previous diagram, you can say the morula and the blastula stage and the formation of the blastomeres. So cleavage begins with the zygote that progresses through a compaction of the morula stage and terminates at the start of the blastocyte, blastula stage. So the first 8 blastomeres that are undifferentiated and have an identical potential in mammals. Thereafter, the blastomere differentiated into the inner and outer cells. This illustration shows that how the cleavage division starts. So this is the formation, the first cleavage division, and then the formation of blastomeres, these 4 blastomeres in the second cleavage division, and then the formation of the morula, in which the outer blastomeres and inner blastomeres you can see, and then the formation of the blastula, blastocyst. Here we can say, why we say it blastocyst, that this cavity is filled with the fluid. So you can see the inner mast cells and the blastoceles, the cavity filled with fluid I was talking about, and then the trophoblast you can see. So this slide show the 2-cell stage, the individual cells, the blastomeres, then the mitotic division occur that maintains the diploid complement, Sometime the phenomena occurs that cells become smaller, and then you can see the blastomeres that are equivalent. So sometime I show you the human embryology diagrams, so it will help you to understand basically that what happens. And this is the 4 cell stage, the second cleavage division. So here in this the 4 equivalent blastomeres, and they are still in the zona pellucida. The you can also see the electron micrograph here in the C. So this is the electron micrograph. Here you can see the 4 cell stage under the electron microscopes also. So this is the 8 cell stage, the third cleavage division. You can see the blastomeres here in this cell stage. So coming to the morula formation, a solid ball of blastomeres within the zona pellucida, typically consisting of 16 to 64 blastomeres at the end. So blastomere become compacted cells on the inside, differentiated from those along the surface of the morula. So the outer blastomeres become flattened and form tight junctions, the reducing the fluid permeability. They develop the capacity to secrete fluid. They are designed to become the trophoblast, which form the chorion and amnion, and these chorion and amnions are the two fatal membranes of the conceptus. The inner blastomeres from gap junctions to maximum the intercellular communication. They designed to become the inner cell mass which form the embryo itself, also its fetal membranes. Coming to the blastocyst or blastula, so it develops during the second week after the fertilization, when the zona pellucida ruptures. So it consists of a large number of blastomeres arranged to form the hollow fluid filled spherical or cylindrical structures. So it contains an inner mass, that is embryoblast, that evident as collection of cell localized inside the one polar end of the blastula. These surface cells of the blastocyst are designed trophoblast. This future chorion formation occurs here in the fetus or conceptus. The cavity of the blastocyst, that is called the blastocele. Eventually the blastocyst attached to or implants within the uterine wall, depending on the species. The formation of 3 germinal layers, the ectoderm, endoderm and the mesoderms. These are the designated primary germ layers because all organs originated from these 3 germinal layers. Ectoderm form the epidermis of the skin, the epithelium of the oral and the nasal cavities, and the nervous systems and the sense organs. So all these things are very important, and you know their importance as you learn about them in the systemic histology. So mesoderm, they form the muscles and the connective tissue. Also the connective tissue include the hard connective tissue, that known as the bone, and the components of the circulatory system, urinary system and reproductive systems. So the endoderm, they form the mucosal epithelium and glands. Sometimes we say the glandular epithelium. Also the respiratory system and the digestive systems. The hollow tubular organs of the digestive systems are also part of that digestive system. Thank you, dear student. It is the end of the today's lecture. In this lecture, we have learned about the fertilization, cleavage, the formation of three germinal layers, the formation of different systems from these three germinal layers. In the next lecture, we will try to cover different aspects of gastrulation and organogenesis. Also, we will discuss the placentation. So maybe this lecture come after your exams. So stay tuned and learn about the remaining part. I am here talking about my students. So dear students and audience, after that we will start the histology series, and hopefully you will like the histology series. So stay tuned and wait for the histology series. Thank you very much. So nice of you. Bye bye. Pakistan. Pahindabad.